Thank you. Really appreciate the opportunity to be here with these other wonderful speakers and experts. And hopefully this talk will build on everything that they've set up so far and give us the opportunity to think about a particular case where our community has worked towards this pathway of reaching alignment with FDA regulators around the use of a biomarker. And also share with you why the accelerated approval pathway is so critical for diseases like neuronopathic MPS diseases. I work with several companies in the space, helping them think through their clinical trials. And just to kind of set the stage, we've heard about accelerated approval, but I thought it might be good to think about why did it come about and why are we talking about it so much now? It began back in 1992 with just FDA regulation, and really that was from the push of HIV advocates and around the HIV/AIDS crisis and the need to urgently accelerate drug development for that disease space. It was quite effective. In about 5 years later, this was codified into law. And then later in 2012, we see the first amendment and adjustment updates to this act, where FDA was then given authorization to require post-marketing studies, to review promotional materials, and to be able to withdraw drugs that were approved on this pathway that didn't ultimately meet post-marketing requirements. Fast forward 2023, another amendment we get where FDA is then allowed to have authorization to specify the timing and the design of these confirmatory studies. And this is probably in response to maybe products that were taking a bit long to complete their confirmatory requirements. So this just allowed more authorization for FDA to do its job and to provide some more procedures and details around how to withdraw drugs. And we are here at 2025 with a lot of changes happening, but the discussion around accelerated approval continues to be at the forefront, and we hear the new FDA commissioner talking about the need to expedite drug development for people with rare and serious diseases, so that's encouraging. Thinking about accelerated approval, and we've heard about this already, but there's really just a few criteria. Your product has to be treating a serious or life-threatening disease. So these are people with severe unmet need. This is not the mass population of people who have allergic rhinitis. We are talking about serious diseases here. They have to determine that the product has an effect on a surrogate endpoint, which we've heard a lot about. And really that key phrase is the endpoint has to be reasonably likely to predict clinical benefit. And they're also taking into account whether there are alternative treatments that can meet the need. So looking at how our disease might fit those criteria, we know neuronopathic MPS diseases are absolutely devastating, and I'll focus on Sanfilippo syndrome here. But children are born fairly typical neurodevelopmental kind of pathway in that early year or two of life, and then start to slow in their developmental gains. They develop severe behavioral disturbances, often diagnosis with comorbid autism, severe sleep issues, sometimes not sleeping for days, and then begin to progress in their regression of skills. You see my daughter Eliza there at 4, not long after she was diagnosed and probably at her developmental peak, but running around wildly, talking, singing, and doing quite well. And then her friend Isabel at age 11, who at that point had already lost most of her independence, was being fed through a G-tube and passed away just a few years later, which is the typical, on average, life expectancy for children with Sanfilippo type A. Along with this comes severe neurodegeneration and loss of brain tissue. Uh-oh. Somebody's iPhone's trying to connect. Sorry. Okay. maybe I need to go back. Let's see. Okay. Sorry. So time is really critical. And of course, we would love to intervene early. We want to prevent all of this from happening. But most of the population doesn't come to diagnosis until age 4 or 5. And at that point, there's already severe neurologic burden of disease. Most studies have focused on a cognitive outcome. And again, if you intervene early, that's quite good. But we don't have the population to enroll in those studies. And even at that point, as Rebecca was saying, you will have to follow kids out for 5-10 years to really see significant difference in their scores. So, this traditional clinical trial paradigm was not working for our disease space. You see all the red. These are lost programs that had been halted, shelved, returned to academia, or companies that had gone out of business trying to stay afloat. So we knew we needed to be thinking about this in a different way. Back to what Rebecca was sharing is, was cognitive outcome the right measure for the whole population? And what do we need to be focusing on? So we conducted a caregiver preference study and have a couple publications that came out of that defining the unmet need. And then further going on to say, what are the priorities of those unmet needs and how can we, kind of do some face validity understanding of whether certain outcome measurement tools align with those and would those be useful for clinical trials. What we heard from families was, yes, there's an understanding of, in a perfect world, we would love our kids to be cognitively normal. I mean, that's a given, but we're in reality and our kids are affected. And so what's important now? Communication is important, and it says speech here, but this is really more global communication. We talked about comfort. You heard from the MSD community that comfort was important, and pain, distress is important across the board for our families, but also things that are really actually quite measurable in a clinical trial, things like being able to continue walking for longer, being able to eat and drink by mouth safely for longer. Those are really key factors. So we kind of have these two cohorts or groups of children. We have very few children who are identified early in life at this pre-symptomatic stage, usually sibling diagnoses. So there's a little number of them that actually could demonstrate the largest magnitude of clinical effect on a cognitive outcome. And then you have 99% of the rest of the population that, frankly, we need to stop ignoring and try to find what is meaningful benefit for them. And we know what is meaningful. The families have told us that. And so beginning to deploy outcome measures that can measure these things. And companies actually have started to look back at the data they collected and try to draw out information on that, which will be really important for long-term studies. Again, in either of these cases, these children have to be followed out 5 to 10 years minimum, which is not tenable for most small biotechs working in ultra-rare disease spaces. So, accelerated approval. Use of a biomarker is the obvious kind of solution to a clinical trial paradigm that was not working. We had some hope when this guidance came out in 2018, which really kind of opened the door for our disease space and diseases like us that were focused on a single enzyme defect with substrate accumulation, saying that measurement of that substrate would be reasonable for accelerated approval consideration. However, just a couple years later, specific to Sanfilippo, that was kind of walked back by the agency, and they indicated that they needed additional evidence to feel confident in the use of that biomarker. It was very disappointing, obviously. It felt like a big blow, but it did give us really something concrete to respond to. And, I would say for patient advocacy groups and academics out there to use those opportunities to really provide additional information that is really so important for the FDA to be able to make good decisions for the patients. our communities kind of coalescing together around this issue really grew over time. Emil Gakis, who's a leader in this space, and many others also kind of helped pull our groups together, and we began working together very actively to get the word out that the accelerated approval pathway is not a lesser pathway. It is a fit-for-purpose pathway for diseases in which traditional drug development is not appropriate. And so writing op-eds, opinion pieces, interviews, really just keeping the conversation at the forefront. We then had the opportunity to be hosted by the Reagan-Udall Foundation for the FDA, which was an incredible forum for broad stakeholder collaboration. And the people pictured here were speakers and panelists, but there were so many others kind of behind the scenes that took place, really helped pull everything together. And so this was an important thing. Companies broke down silos and shared information, and that was another key and unusual thing that happened here. But as we were thinking about how to use that time that we had most effectively, we really had to think about what were the gaps we needed to fill in. And so thinking through, as you're working towards a biomarker for use as a surrogate endpoint, what questions do the regulators have? Some of these are very, very reasonable questions, and it's really kind of on us to put that information together in a cohesive and clear way that the message can be shared effectively. So these are just kind of a few of our gap analysis. Yours may look a bit different for your disease space, but always important to pull in the real world perspective, hearing from patient advocates and families, what are the actual implications of the disease, of not being able to move forward with drug development and have that being stalled, seeing programs removed and people discontinued from therapies that were seemingly effective. Peter Marks also spoke and opened that meeting where he spoke about the importance of accelerated approval for just this type of diseases. And I think it's important to remember that accelerated approval is not intended to get it right 100% of the time. It's intended to meet the need of these really horrific diseases and go with what the science leads us to, knowing that we will have to confirm clinical endpoints down the road. But he also shared about, if you have a biomarker that seems plausible, that seems like there's correlation, that's wonderful, but you really have to establish that the assay that measures that biomarker works, needing to be accurate and precise, as Dr. Bryan spoke about earlier, and in the context of use, like what sample type are you using? For us, it was spinal fluid, so does that assay work well in that sample type? But that formal qualification is really something that's probably not fit for, rare disease specific programs or necessary. In our workshop, we shared about how assays improved over time, and that was an important question to answer for FDA because there were some probably misinterpretations of clinical trial outcomes along the way where older assays were used. Total GAG, non-specific assays, whereas now we have the opportunity to use the most advanced technology using mass spectrometry that, as Rebecca pointed out, can very specifically delineate the GAG fragment that is associated with a specific enzyme deficiency. And there are different methods to use this, but they both correlate. This is an example of where there was a misleading clinical trial outcome interpretation. And Dr. Munzer presented this from the Hunter's syndrome community. And this was an intrathecal enzyme replacement where they initially measured total GAG through a thrombin assay, which showed about an 80% reduction in spinal fluid total GAG. They did not meet their primary cognitive endpoint. Later analysis of those samples with newer technology showed that indeed there was only a 30% reduction in spinal fluid heparan sulfate. And so you really have to reconsider what those clinical endpoints mean, and these children are likely, significantly underdosed, and so this trial was not set up for success because the assay was not precise enough. This same thing happened in a similar program for Sanfilippo. Another question was around, does your biomarker reflect your tissue? So if you have a fluid biomarker like spinal fluid, is it reflecting brain accumulation? And Dr. Dixon designed a very elegant study where she restricted expression of NAGLU to either brain neurons or to the periphery, and was able to really delineate the correlation of CSF and brain HS accumulation being separate from serum accumulation, and vice versa. And then Dr. Matthew Ellenwood also shared this really continuum and broad perspective that heparan sulfate in the spinal fluid and in the brain correlates across species, across MPS types, across MSD, as we've heard, that heparan sulfate fragments are indeed toxic and, similar to PKU, are poisonous to the nervous system. And you'll also see here that it is treatment responsive. So kind of looking at that bigger picture of the animal data that you have, pulling all that together, also looking at the other biomarkers that surround that, and Rebecca talked about that some too. We were proposing the CSF-HS, which is the most primary biomarker, and again, disease-causing substance. But there are other biomarkers downstream that can be information can be provided to kind of help support the context of that heparan sulfate, and then you see much later down the clinical outcomes with cognition. We were able to hear some emerging clinical data, which again starts to establish those correlations between heparan sulfate levels and human clinical outcomes. Again, these trials were focused primarily on cognition, and you see reduction in HS associated with divergence from natural history and raw cognitive scores, and this is the Ultragenyx product presented by Dr. Lau. But those children are followed out till age 5, treated before or right around a mean of 20 months of age. So you can see that very long time span needed to reach the clinical outcome. And then just to follow on the story this year, additional data as that continues to mature shows further divergence. We do not see yet quite divergence in gross motor, but that is expected. So that helps us say, yes, all of this is appropriate because gross motor declines later. And then another program using lentiviral autologous stem cell transplant. This is an Orchard program showing near normalization of heparan sulfate and early positive developmental data. We left that meeting feeling great, but this was not a situation in which we got an answer from FDA. That's not how those workshops are set up. So we wanted to really provide a enduring reference, not only for our community, but for FDA and anyone else entering the space, that indeed the preponderance of evidence supported the use of CSF-HS as a measure that is reasonably likely to predict later clinical benefit. And good news, since then, we have really seen that movement come along, and these 4 programs have all announced that they've reached alignment with FDA on the use of the biomarker for accelerated approval. Several of these have actually already filed their BLA at this point. So really exciting progress that would not have been possible without the acceptance of this biomarker. And just to end, to thank so many people who have been involved in the advocacy and the hard work to bring forward the science and put it together to help us reach where we are now and help our kids be healthy. So thank you.
Time for a few questions.
I have a question. Kara, so since there is no formal or regulated role of patient advocacy group in the drug development and approval process, and given your involvement and kind of success and hearing your voice as a community in this story, would you have any like a model, or maybe you share some thoughts, suggestions on that other patient advocacy groups can use to build this relationship and also contribute to the process in other cases?
Yes. Well, I think you also heard from Rebecca how her community is kind of coming together, and I think you have to have industry partners also who are willing to share, are willing to kind of break down some of those silos, because if we don't understand what the questions are from regulators, we're just guessing, and we can't really address them. And so that's the critical part, is people coming together around a specific issue that really affects us all in the disease space. Yeah.
Great talk, Kara. One thing I just wanted to applaud you guys for taking the time to publish the findings that you come up with as a collaborative group, and even after the workshop, making sure you put that consensus paper out. I think that's really helpful to the community. My question is that opportunity that you had to get in front of the regulatory body with that collective group, how did that come about, and is that an option for other patient advocacy groups that might want that kind of platform?
Yeah, thank you. Well, I will say we had other opportunities to meet with FDA leadership prior, and some of those were facilitated by industry partners, which were broad and not specific to any kind of product, but really around the topic of accelerated approval. However, the Reagan-Udall Foundation does host workshops like these. I think there were 4 industry partners who sponsored that, the work of the Reagan-Udall to host that financially. But and so that is open, and I would encourage folks to reach out to Susan at the Reagan-Udall Foundation, or there are other groups who can host such events. But I think what was nice there is that their connection and collaboration with FDA allowed more people to be in the room together.
I think we have time for 2 more questions.
Hi, Emma James, Encoded Therapeutics. Thank you. This is a great talk. I just wondered if you had any feedback or experience from payers or HTA bodies around their thoughts on acceptability of biomarkers or accelerated approval in terms of then reimbursing the product.
I think this is more of an issue for common diseases. And I think for these devastating ultra-rare diseases, the conversation's a bit different. Accelerated approval based on a biomarker or an intermediate clinical endpoint is an approval just like any other approval. It meets regulatory standards for safety and efficacy with that commitment for the longer-term clinical outcomes. So I think, it is important that we're all kind of sharing that message that accelerated approval is approval. It is not a lesser approval, but it is a fit-for-purpose approval for the right product and disease. So in our disease space, we are just kind of getting to the point of, a BLA's filed, it's under review, and I guess we'll see about the HTA payers, but I expect the conversation is different around these orphan devastating diseases.
Okay, thank you.
Thank you.
And 1 more question.
Thank you for your presentation. It's really encouraging. Regarding the accelerated approval process, at what point, and maybe if you could just provide a little bit of insight into sort of the conversation that took place about when the right time is to pursue an accelerated pathway in the context of a surrogate endpoint, because you may not have perfect information, but you have leading information that can sort of help you draw a conclusion. How do you know when you have a large enough N, or you have a large enough data set to be able to go back to the regulators and say, hey, look at this, this is very interesting, give us this pathway and we can turn it into something real?
Well, everything's driven by, the unmet need, right? So I think your kind of impetus and urgency to move forward very much depends on the disease you're talking about, and your ability to enroll larger numbers in clinical trials. I mean, I think the adaptive trial designs where you're not necessarily doing separate studies, but a continuation of kind of a longer adaptive study is really well suited for this type of disease space. And as FDA says, talk to us early and often. And I think, the START program and some other programs that they have like that, I hope that we'll continue to see building on that so there can be more rapid, real-time interaction. Bandwidth is always the challenge, right? But as soon as you start to see some of those correlations bear out, or you have a preponderance of evidence to say this is an appropriate biomarker you need to start to, I don't think it's ever too early to do that, because it's going to impact how you design your trial and move forward.
Thank you.
Thank you, Dr. O'Neill.
Thank you.